Okay, I think we are ready to officially start. I would like to welcome all of you to the 25th in a series of Zoom lectures sponsored by Congregation KINS. I see some new names out there, so I will mention that our conferences are all based on a STEM topic: science, technology, engineering, mathematics, and we have added an extra M for medicine, psychology. I am going to be the speaker today, and the topic is a very technical one, but related to medicine and chemistry. And I will promise you that the chemistry will be low level so that you don't have to think back to your high school and college days for chemistry. The topic today is Yad Hashem, the hand of God and the discovery of medical miracles. And I will encourage you, if you have any questions at any point in time, please don't hesitate to interrupt me. Just unmute yourself and ask your questions. So I'm going to share my screen with you. Let's see, slideshow. And we're going to go ahead and get started. Every day when we say Shemon Esrei, we thank Hashem. We thank Hashem for giving us knowledge and understanding and the ability to discern things and to learn new things. And two of my favorite psukim in all of the Tehillim are the following: Ma gadlu masecha Hashem, how great are your works, and also Ma rabu masecha Hashem, again, how great are your works. And the Chachamim have actually gone a long way to describing what this means, but just to give you a very simple example, Ma gadlu refers to something large in size, and Ma rabu refers to something large in number. A real crude example might be a beautiful 10-mile long beach with white sand. That's a large thing. The small thing might be the gazillions of beautiful little grains of white sand that make up that very large thing. And in any case, all of God's works are done with great thought, great depth, and great wisdom. The next few slides are just going to be the Torah part of this, and I'm not going to say that Hashem created this miracle on every single one of the examples that I give to you. But fortunately, Hashem has made all of us intelligent and curious, inquisitive, tenacious, and very often there are people who have sixth senses about things and make discoveries that might not even be logical. We often hear people talk about, oh, that's a coincidence, but we Jews really don't believe too much in those kinds of coincidences. We believe that Hashem controls everything, even the smallest things in our lives, and he's the one who makes things happen. And fortunately for us, there are people who think outside the box, and they say things like, well, what if this happened, or what if that happened? One of the things I will freely admit is I don't like snakes. I especially don't like poisonous snakes, and I am truly afraid of them. And I think most people feel the same way. But thank God there are people who said, what if? What if I took that snake venom and examined it, look at the chemical composition? Maybe there's some medical value that's in there. And that kind of thinking, what if, what could happen, has saved many lives and truly has prevented a great deal of human suffering. So I'm going to show you a few stories and small vignettes of Hashem's control over this world. And I think that you'll see here that some of these things appear to be fortuitous, but we know who's responsible for these medical discoveries. There's something that no one really knows why these things happened, but they did. 3,500 years ago, who thought of taking the bark from a tree or drying myrtle leaves and giving them to people to alleviate headaches and other kinds of pains? There's no logic to that whatsoever. I can't imagine chopping off bark from a tree and chewing on it, hoping my headache would go away. But someone had the intuition to do that. And in fact, take a look at the second point here. The ancient Egyptians used to take moldy bread, moisten it, and fasten it to a cut or a scrape to prevent infections. And we're going to see in a few minutes exactly why that works. Only God knows why they thought of it, but it actually has to do with something that is part of our everyday lives. We'll see it shortly. We all know that Hashem has created plants and animals, fish and humans, and there, of course, is their physical beauty. We all know about beautiful plants, beautiful trees, beautiful flowers, beautiful animals, beautiful fish, beautiful humans. So we're very aware of their physical beauty, but there's something that I like to call their hidden beauty. And that hidden beauty is actually on a molecular level. It's something we can't see, and it's therefore not evident. And it takes someone who has a creative mind to help to determine if it's even there. So we're going to take a look at the molecular level of everyday things and see how they have really helped mankind to stay healthy. One of the most important takeaways from what you're going to see in the next half hour or so is that there are many very, very toxic substances in this world. There are some in our front yards and some in our backyards, and they're all over the place. And one of the things we have learned, perhaps the hard way, is that while these substances are very toxic in very small quantities, they're often life-saving. And if you take 100 milligrams of some of these toxic substances, you would die. But if you take 100 micrograms, it might save your life. And you're going to see this as a recurrent theme over the little stories that I'm going to relate to you shortly. Anybody know what this plant is? Anybody recognize the beautiful flowers? This is a flower called Foxglove. You know, in one of his very long poems, John Keats started it by saying, "A thing of beauty is a joy forever." And that, of course, is certainly true with all the beautiful things in our lives. But here, we can say both fortunately and unfortunately regarding foxglove. The unfortunate thing is that every part of that plant is toxic. The leaves, the petals, the berries, the stems, the roots, every part of it is toxic. And the fortunate thing is that there was someone who had the inspiration to evaluate the foxglove and say, what's in there that's causing that toxicity? And maybe there's something useful we can do with it. And so the original observation came from accidental overdoses. Mostly they were in children who just came over and ate some of the flowers. And in fact, there was one case I read about this week in the literature where an adult heard about the toxicity, a woman, and tried to kill herself by eating foxgloves. I don't know whether she thought it was fortunate or not, but someone actually ended up saving her life. And so the net effect of consuming foxglove is that it is a cardiac poison. It will slow the heartbeat. It can cause irregular heartbeat. And if you consumed enough of it, it would lead to death. So Hashem made sure that the right researcher with the right talent looked into this poisoning, this cardiac poisoning, and chemically analyzed some of the components that were in there. And he isolated a compound that you might have heard of called digitalis. And digitalis was chemically converted into what is now a very, very commonly used drug called digoxin. I've taken it myself before. And digoxin is such a powerful drug that the amount that an adult would require to control cardiac rhythm and to increase cardiac output is measured in micrograms. If you imagine what 1 grain of salt looks like, that's way larger than the dose of digoxin that a human would take. And milligrams would certainly poison and probably even lead to death. There are other compounds also besides digoxin that were created from this plant. One of them is called digitoxin and one is digitoxygen, and I'm sure there are some others. If you have ever been to South Carolina or Florida, This is a plant that is very, very common. I grew up in Charleston, and all along the sides of the road, we see these plants, beautiful flowers, and they're called oleanders. And the cardiac toxicity is so high that if you eat one leaf, and I'll point to one of these leaves here, they're usually 5 or 6 inches long for an adult plant. If you were to eat one leaf, the chance of it's being fatal is very high. It's just such a good poison. They're actually called cardiac glycosides, and the two components in there that are toxic have these names: oleanderin, named after Oleander, of course, and nerine, named after someone named Neri, who was one of the researchers with this plant. And while the molecular structures of these two are totally different than the digoxin kinds of molecules, the physiological effects are almost identical. They're both cardiac poisons. They both slow heartbeats and cause irregularities, and certainly could kill people. And from a very technical standpoint, there is a metabolic process called the sodium potassium pump. Everybody has it going on in their heart all day, every day. It's a chemical mechanism for pushing potassium into the heart and taking sodium out. And that helps to keep the heartbeat very, very regular and very strong. Now, Hashem again made sure that the right person was there with the right amount of curiosity and with the chemical talent that was necessary to evaluate this plant. And while I won't go into the chemical details, people harvest the leaves and the flowers of this plant, and there are now numerous medications that are used for all of these 10 things down here on the bottom: heart disease, asthma, epilepsy, cancer, and a whole number of other things, even including venereal diseases. All these came from a plant which is basically toxic. Anybody know what this is? Recognize this little guy? This is a Gila monster, and Gila monsters in their venom and their saliva have a protein called exendin-4. I really don't know what the structural formula is, but proteins are typically very large molecules with molecular weights up in the many thousands. Our body has something called homeostasis. This is the condition of having everything work properly all the time. And what this drug does is that it causes homeostasis for glucose levels. In fact, the drug is called Byetta, B-Y-E-T-T-A, and it's one of many new drugs that help to regulate glucose metabolism in type 2 diabetes, not for type 1, which is mostly a childhood disease, but type 2. And so there are people who actually raise Gila monsters simply to collect their venom and their saliva to create this drug. How about snakes? There are many successful drugs on the market today. And here's a short story of one of them. You can see a picture here of a very large snake. One of the most successful drugs on the market is called Captopril. It's used for people who have hypertension and renal or kidney disease, usually resulting from diabetes. And this drug came from a snake venom. And in fact, I'm not sure it's still coming from there. It might be synthesized chemically now, I'm not sure. But this is called an ACE inhibitor, an angiotensin converting enzyme. And it's used to treat all of these things here: hypertension, heart failure, and so on. And it has literally saved many, many lives. It is one of the most successful drugs in the world market. Some scientists in Taiwan were working with a pit viper called Wagler's pit viper, and they noticed that when this pit viper bit an animal, it would inject this venom, and the animals would literally bleed to death internally. And we might think, okay, that's interesting scientifically, but someone came along and thought, well, that might be a very good blood thinner. We know about things like heparin and Coumadin and Xarelto and stuff like that. And this is actually currently being evaluated to see if it might be better than the current anti-clotting agents, maybe better in the sense that it doesn't cause the very significant bleeding that some of these other drugs do. Also, it's very common today when people get snake bites, they're taken to the emergency room and they're given an antidote for the snake bite. Where did that antidote come from? Poisonous snakes today are actually milked for their venom just routinely. That venom is collected and chemically treated to convert it into an antidote to save people's lives from the snake bites. This is an animal I had never heard of before. It's an underwater animal, and there are hundreds of species of them, and every one of them has a beautiful shell structure. Now, if you look carefully here, there's a light-colored protuberance that's coming out, and at the end of this is a pointed piece, and the venom comes out of this tube. And when it comes near a fish, it pokes the fish with this venom that paralyzes the fish, and then this large tube up here comes down and suctions out the tissue from the fish and provides food for this animal. Now, again, you might think that's scientifically interesting, but somebody said, wow, you've got a venom that causes paralysis. We might be able to do something with that. This ended up being a drug on the American market called Prialt, and it is used to reduce the suffering of chronic and sometimes intractable pain that simply cannot be relieved by anything else except cutting of nerves. And so this is a very valuable drug. It's used pretty commonly all over the world. And just think where it came from, a cone snail. Pretty amazing. The story of penicillin is truly amazing. This was in the 1940s that penicillin was introduced as a drug on the American market, in fact, on the world market. And prior to that time, when people had bacterial infections, unfortunately, they were put in the hospital. The doctors had nothing to treat them with, and most of them simply died. The doctors were simply hoping that there would be a cure. And there was a cure starting in the 1940s. But let's back up about 10 years. This truly is an amazing story. And this took place, the beginning of it, in the United Kingdom, but the United States ended up being the one that developed the large-scale production, enough for wartime, because this was right during World War II, or right in the beginning of it, and no one had ever discovered any antibiotics before. And what are antibiotics? They're really substances that are secreted by bacteria and molds and fungus to prevent other microbes from attacking them and killing them. And so it turns out that this phenomenon had been known for centuries. I mentioned earlier about the Egyptians. Look what they did. They took moldy bread and slapped it onto infected wounds to cure the wounds, and apparently it worked pretty well. So in 1928, a chemist by the name of Alexander Fleming lived in London, went on a holiday, but before he did so, he had a bunch of petri dishes and he was culturing colonies of streptococcal bacteria. They cause all kinds of terror in people, boils and sore throats, terrible abscesses that require an antibiotic to cure them. He went on vacation, came back a week later, and lucky for him, he left a window open in that room where he had his petri dishes. Into that window came a mold, a particular mold called Penicillin, and the mold settled on that one petri dish. And I want to show you what happened. You know, they say the internet is the source of all knowledge. And this is Alexander Fleming in his lab. You can see the petri dishes back here. And this is an actual photograph of the colonies of streptococcal bacteria here. Here is the mold. And you can see around it a pretty clear area all around here where there are no colonies. There are a few teeny ones, but those are what are called lysed, L-Y-S-E-D, lysed colonies and bacteria. That means that their cell walls are ruptured and they are either dead or dying. And so this mold was secreting something that was preventing bacterial growth. And Dr. Fleming in he called this mold juice, and he did a lot of experiments with it in that first year, and he found that it was capable of stopping things like streptococcal infections and meningococcus, which caused meningitis and diphtheria. And he tried to isolate pure penicillin. That was the name he decided to give it to him. He really didn't have much luck. He developed some crude material, but as scientists like to do, and 1 year later, he published in the British Journal about his research. And he didn't really describe anything that could be done with it. He gave a hint, just a passing reference, that maybe penicillin might have therapeutic benefits. And then nothing happened for almost 10 years. 1939, 10 years later, researchers at Oxford University decided to have a good look at the penicillium mold, and they actually ended up purifying penicillin. This was right around wartime, and they collected enough of the penicillin that they thought they would have enough to at least treat one patient. Along comes an emergency. Here's a 43-year-old policeman who was trimming his rose bushes, and a thorn on a rose bush simply scratched his cheek right near his mouth, and he developed life-threatening infection with abscesses that affected a lot of the parts of his body, his eyes, his face, his lungs. They debated whether to inject this because they didn't know if they had enough penicillin. No one knew what the correct dose was, but they decided we can't let him die. Let's give it a shot. So they literally gave him a shot. They gave him an injection of the penicillin, and they gave it over several days, and he had a remarkable recovery. And then here was the problem. They ran out of penicillin, and they hadn't given to him enough of it and or long enough. And unfortunately, he died a few days later. Still, they learned a lot from that first experiment, and they made sure that they had lots and lots of penicillin on board before they had anyone else injected. And so they then prepared enough for the troops on the battlefield. And very interesting that Illinois played a big part in the production of penicillin in the United States, because in England, they couldn't do it during wartime. They just couldn't put the resources together, the people, the labs. And so the USDA, the Department of Agriculture, got a hold of its research lab in Peoria, Illinois, and they came to the rescue. They developed some innovative chemistry steps and scale-up steps, and they made lots and lots of penicillin possible. And so Fleming and two of his colleagues who did the research, they were the Oxford people, were awarded the Nobel Prize back in 1945. There's a drug now called Taxol. It is the most well-known cancer drug in the US that comes from totally natural sources. There's nothing synthetic about this. And this came from research on the bark of a tree. People were taking barks of trees and leaves and stems and roots of everything imaginable, just evaluating them. Maybe there's something there that has medical value. And so this drug ended up being approved, as we'll see shortly, for breast cancer and lung and ovarian. And ovarian is particularly difficult to treat, and also something called Kaposi sarcoma. Now, this all started with the Pacific yew bark back 60 years ago, and it was discovered that that had cytotoxic activity, and they finally prepared and isolated this drug. In 1977, the National Cancer Institute confirmed that it had anti-tumor activity in melanoma and breast tumors and colon cancers. And little plug here for Yeshiva University, Dr. Susan Horowitz from Albert Einstein Medical College won a grant from NCI to study Taxol's mechanism of action, which she did. She determined how Taxol was able to kill the cancer cells for those various diseases. A company called Polysciences did a large-scale production, and then they started looking at a different kind of yew tree. It's the one I have in my front yard. You might have it in your front yard. And this is a different version of the genus Taxus. And it's so common and so available and so cheap that became the source for Taxol. Phase 1 clinical trials began in 1984, and when Johns Hopkins University Hospital in Baltimore reported that the drug created partial or complete remissions in a significant percentage, 30% of people with ovarian cancer, that was pretty miraculous. There was nothing good for ovarian cancer prior to this. And so in 1992, the FDA approved it for ovarian, 1994 for breast cancer. And today there are clinical trials with Taxol for many other kinds of cancers. And it's interesting that this has historically been the most lucrative drug ever for cancer, the most best-selling drug of all. Annual sales have averaged anywhere from $1 to $2 billion per year just for that one drug alone. Vitamin C. Now, we are all very familiar with vitamin C, and the historical perspective on this is really pretty interesting. You know, we all today can buy any fruit and vegetable we want. You want watermelon in December? Go to Jewel. Everything we need is available. But that was not true centuries ago. And in fact, sailors who were on long trips that lasted for months often ended up with swollen and bleeding gums. Their teeth often fell out. They had subdermal hemorrhages, and their wounds took forever to heal. Toward the end of the 15th century, it's interesting that the people in Denmark, mariners, had somehow figured out that there was some nutritional thing in citrus fruits that was preventing this scurvy from happening. And so they included lemons and oranges in their marine stores. It took the British about 250 to 300 more years to make that discovery. And so they were still suffering around almost to the year 1800. A Scottish physician, Dr. Lind, knew about the preventive powers of citrus fruit and lemon juice. He's a naval surgeon, and he recommended, and they did this, lime juice to go on every single British ship, and that actually eliminated scurvy in the entire British fleet. Took a while, took a few years, but completely eliminated it. Here's an investigative British physician, a Dr. Stark, and he decided to be the guinea pig himself. So he put himself on a really terrible diet where all he ate was meat and starch. He ate no vegetables, no fruit, and he not only developed a really bad case of scurvy, but he died from it, unfortunately. So it was, I guess, some living proof that these citrus fruits are a lifesaver. Along came, I don't know if you can see the picture on the right. It's a picture of Dr. St. George. He's the Hungarian-born researcher, and he took a look at orange juice and lime juice and lemon juice and said, what is in there that is preventing the scurvy? And being a good chemist, he finally isolated and identified ascorbic acid. I don't know if you can see the image on the bottom right, but there's a molecular formula for there, pretty irrelevant, really. And this was back in the 1930s. And he also, and this took some real genius, he also learned what the mechanism was, the metabolic mechanism on a molecular basis that let our cells use vitamin C to metabolize fats and proteins and carbohydrates. And so he actually won the Nobel Prize for his terrific discovery of vitamin C. Steroids, we're all pretty familiar with steroids. They all happen to be fat soluble or lipid soluble compounds, and we're really familiar with many of them. We've all heard of cholesterol, progesterone, testosterone, the male hormone estrogen, the female hormone estradiol, another chemical that's used medically, and of course, cortisone. These all have a very similar structure, the steroids. And initially, this was back in the 1930s, some of the chemists used to call it the decade of the sex hormones because that was when they were determining the molecular structures of these. And while the last line talks about oral contraceptives in 1951, that is not why they did this. In fact, this guy, Russell Marker, was the first one to find a practical synthesis for progesterone. Why did he want that? As you'll see in the next slide, progesterone was used to help prevent spontaneous abortions, miscarriages in women. And look at where he got his starting material from. Who would have thought that a Mexican yam contains a precursor to progesterone, but it happens to have a steroid-like molecule? I have no idea how he even thought to go look for Mexican yams, unless someone may have reported that in the literature before. But he took this and he developed the product, the precursor progesterone, which happens to be a precursor for cortisone. And so the attention was really first on progesterone for preventing miscarriages. And the cost of this drug was enormous back then. But once the Mexicans got involved with those yams and creating steroids, the cost went way down. This guy, Dr. Marker, was a professor in Pennsylvania. He actually produced progesterone from a lot of things, one of which was sarsaparilla. And a couple of years later, he saw in a journal a picture of a plant, and the plant was known to have molecules of steroid similar to the one that was in that yam. And so he went to Veracruz, Mexico. He purchased 10 tons, 20,000 pounds of this root, and he isolated 3 kilos of progesterone from it. That's 6.6 pounds out of 20,000. That's 3 parts per 10,000 of that root. That's a tiny fraction. 3 10,000ths of it was progesterone, and the other 9,997 10,000ths of it was something else. And this was valued at $240,000. Now consider, that's back in 1941. That was a huge amount of money back then. Progesterone, 6 pounds, it would certainly not cost that much to produce now. Now, cortisone, for many people, as it says here, life would be unimaginable without corticosteroids. Many, many people use it for treating arthritis, asthma, autoimmune diseases. There are all kinds of creams for skin conditions, certain kinds of cancer, and many of them today are available mostly for skin use as over-the-counter treatments. I don't think you can buy cortisone over the counter, but there are corticosteroids that are available. And the big leader was the Upjohn Company up in Michigan, and their chemists came up with some amazing and innovative ways to perform the right kind of chemistry and improve the manufacturing process. And they became world famous and the masters of steroid chemistry. And the breakthrough that occurred, I can't see that date, but probably in the 1940s or so, that was a milestone in their journey toward producing the next generation of steroids. Now, cortisone was first actually discovered by the Mayo Clinic in 1929, but it took until 1949 for anyone to make enough of it to be able to use it. But there was some use between 1929 and 49, and they found dramatic improvement in people who had rheumatoid arthritis. Now, here's why there wasn't much of it available. They were producing cortisone by going to animal slaughterhouses and buying the adrenal glands of animals. The larger the animal, the better, because the larger the adrenal gland. But that was a very painstaking, very expensive way to make cortisone, because the adrenal gland was small to start with. Adrenal glands in humans are about the size of a pea, or maybe a large bean. And so they needed thousands and thousands of adrenal glands. So this was a very expensive operation. And then along came Upjohn, and they created a way to make cortisone commercially, tons of it. And this, in fact, is a picture of the molecule. And the reason I'm showing you this is not to show you the molecule, but to show you something chemically pretty interesting. This oxygen right here is the total necessity for this molecule to work. If you took this oxygen out and either eliminated it or replaced it with anything else, cortisone would not work. So that one oxygen is a total requirement for it to maintain its biological activity. It's an amazing drug. I had never heard of it before, but you'll see shortly how amazing this was. The drug is called Ivermectin, and as it says here, it would have been so easy to have missed the opportunity to find this drug. A soil sample from Japan is suffering in Africa, and here's what happened. A microbiologist in Japan was looking for new sources of drugs, and so he collected thousands of soil samples from all over Japan. And he found one that was a bacterium found near a golf course. And he reported in the medical literature that there's a compound generated by that bacterium that impedes the activity of earthworms. Now, that might sound like, so what? But a team of scientists in the United States read about this, and they hypothesized that maybe that would be a good thing to use on much smaller worms that cause all kinds of things, heartworms and tapeworms and other things like that. And so they developed this into a drug they called ivermectin. And here's the amazing thing. It kills all kinds of parasites in pets and farm animals. It prevents billions of dollars worth of losses worldwide. And just think, it came from the discovery of bacteria on a golf course. And it also works in humans for preventing all kinds of parasitic diseases. In particular, we're going to talk a little bit about river blindness in some of the poorest countries on Earth, like in Africa, where they don't have clean water. They take water out of rivers and they drink it. And there are teeny little worms that lay their eggs and create teeny little larvae inside their bodies that cause all kinds of terrible things, and one of which is called blindness. It produces so much scar tissue in the eye, creates blindness. And here's a wonderful thing. This is a case of people doing the right thing. A cast of thousands, there were scientists and medical doctors and participants who are willing to be studied to see if this ivermectin drug worked in them. And now look at this. The company that developed this, along with the research institute that started the chemistry, produced tons of this and gave it away for free to prevent this terrible river eye disease, river blindness. It's truly been proved to be one of the most extraordinary things for parasitic disease. And Merck Labs was the first one to actually produce this drug. And then I believe it talks about, actually, I think Merck was the one who actually produced it for commercial use. And in fact, look what it does. It prevents all these kinds of things in parasites in livestock, mites and ticks and bot flies. And it works in all of these kinds of animals here, all of which, we don't eat all of them, but people who have their dogs, for example, don't realize that they're using ivermectin, but they buy something called Heartguard. And Heartguard is ivermectin, and it prevents heartworm in dogs. And this veterinary medicine is the top selling in the world. Over $1 billion spent on ivermectin just to save the lives of dogs from heartworms. And of course, the people who developed this Merck scientist received the Nobel Prize in 2015. Okay, just want to show you one or two more slides, then I'll be happy to take any questions you might have. In the news, on CNN, on every news station, there's been a lot of discussion about the use of antibodies to not just treat cancer, but to treat COVID-19 patients. We're hearing about antibody therapy all the time. And it's actually well known that former President Trump was treated with an antibody preparation when he came down with COVID. And so one of the clever ideas that started back in the early 1990s, so we're going back late 80s, early 90s, going back 30 years, a nice nice Jewish boy named David Goldenberg was one of the first ones to report the use of radioactive antibodies to treat diseases. Turned out he had very limited success because he was using what we call an intact or a whole antibody, molecular weight maybe 300,000. It really didn't work very well. And some other investigators years later discovered that if you take that antibody and break it down into one of the several fragments, the fragments work much better. And so what we're actually doing is taking the antibody with a huge molecular weight, hooking onto it an isotope, which, as the slide says, just goes along for the ride. It's just hooked on. And so the antibody targets the tumor, and the isotope delivers a tumoricidal dose of radiation. For example, in 2001, there was a drug called Zevalin that was approved for non-Hodgkin's lymphoma. Anybody who had lymphoma and whose B-cell lymphoma was positive for an antigen called CD20 could be treated with this radioactive antibody. The antibody would bind to that CD20 receptor, and fortunately, 85% of all B-cell lymphomas had that receptor. And so this caused a partial or complete remission in 80% of all patients who were treated. Pretty remarkable. It's about a 30% cure rate and about an 80% total rate. And very finally, this has to do not with treating humans, but with pyrogen testing of drug products. It is illegal to inject a drug product that is pyrogenic. Pyrogenic means it could cause a fever. Now, mostly if it happens, most of the time, people don't even know it unless you're monitoring their temperature. Sometimes it spikes a fever that causes discomfort, but today that never happens because every injectable drug has to undergo what's called pyrogen testing. Now, this is truly Yad Hashem. There is an animal called a horseshoe crab, and I remember growing up in South Carolina, we used to go to the beach, and geniuses that we were, we used to have contests to see who could throw the horseshoe crabs out farthest back into the ocean. These creatures are pretty large. They're with their horn on them, and I'll show you a photo in a moment. They're almost 2 feet long. And it turns out that the blood of a horseshoe crab gels when it comes in contact with bacterial endotoxin. That's what spikes the fevers. And so here's a picture of the horseshoe crab. It's got a big spike on the edge over there. And about 60 years ago, this scientist, Dr. Bang, and another nice Jewish boy, Jack Levin, actually developed a laboratory test called the Lal test. The animal is called Limulus polyphemus, and we call this the Lal test. It was commercialized first in the United States. I've performed this test myself many times on blood products. And so that was discovered truly. Who would have thought of testing a horseshoe crab for pyrogens? But someone had the intuition to do that, and we're very grateful. So I think that's a very comprehensive overview of some of the modern medical miracles that come from the plant and animal world, that Hashem has guided us to discover them, for which we, of course, are very, very thankful. If anyone has a question, please feel free to unmute yourself. Anyone?
Dad, I put a question in the chat. It looks like a lot of these medications take years, decades to bring to fruition. What do you know, and what have we learned from the sort of, talk about Yad Hashem, the miraculous speedy development of effective COVID-19 vaccines in under a year, way under a year? Is the pace of drug development, because of what we learn in COVID, now going to speed up? Will we always be waiting years and years and years for some of these therapies?
Well, I think the waiting years and years is over. We have become so sophisticated in chemistry of preparing drugs. It's amazing. Think about how amazing it was that COVID vaccine was approved in 6 months or so, when prior to this, the FDA would never release anything without 3, 4, 5 years of testing. I think that we have such wonderful test equipment now with MRI equipment and infrared spectrum and mass spectrometry and other wonderful modalities that let us do things nobody could do back in the day when these other things were investigated. And today we can find a chemical that's in one of these plants or animals that I talked about earlier. And once we can identify it, the chance of going into the lab and making it synthetically is really very significant, so that we don't always have to deal with animals and collecting venom from them and things like that. Sometimes we can manufacture that drug ourselves. So yes, that's a good question. And I think that because of modern science, nothing will take years and years anymore. I shouldn't say nothing, but most things wouldn't. Anyone else? If not, then I'm going to mention one more thing here. I want to share one thing on my screen. And here's what's coming up. Next week, Dr. Jonathan Greer is going to give a presentation on the view of science and Judaism on free will. I will not be here. I'll be traveling and on an airplane at that moment in time, and Rabbi Bressler will be running the show, introducing Dr. Greer and making sure all the electronics works okay. I have about a 90% commitment from Rabbi Eisenberg. He is a mathematician, and he's presenting on some fascinating stuff on pi, as in 3.1415916 pi. Rabbi Levi Mostovsky is going to talk about Hashem's miracles, but not the topics that I spoke about today. And Howard has gotten for us as a speaker, Rabbi Moshe Tarragon, who is in Israel. It's a wonderful speaker. And I don't have his topic yet, but hopefully that should be coming sometime soon. We have over a month anyway, or about a month. And then we will not be meeting on March the 30th because that's still Pesach. So one more shot for any questions? Okay, if not, let me stop the sharing. And good, I want to thank you all for your attention, and hopefully you'll sign in next week, and look forward to seeing you soon. Have a wonderful day and a wonderful week.
Yasher Koach.
Thank you.